Hello friends, I am Suchi. Welcome to my YouTube channel. In this video, we will discuss about genetic code. Most of the living organism contains DNA as their genetical material. This DNA inherits from one generation to next generation. Whatever the characteristic one person have or one living organism have, that will inherit from it to its offspring through this DNA. And DNA expresses its character through protein. During transcription process, whatever the information present in DNA, that will get transcribed to mRNA. So during transcription process, from DNA, mRNA will get synthesized by using one strand of DNA, and from mRNA protein will get synthesized. Whatever the information present in DNA, that will express through this protein. It contains adenine, guanine, cytosine and thymine as its nucleotide. It means these are the four alphabets present in DNA. By using these four alphabets, we can write sequence of DNA. Similarly, protein made up of 20 different essential amino acid. It means 20 different essential amino acid are the alphabet to express or to write protein. But during protein synthesis, DNA express its character or properties in the form of protein. It means nucleotide, which are the alphabet present in DNA, it should convert in such a way that we can write this amino acid by using this nucleotide or by using this alphabet, four alphabet. So how it will be possible? By using this four alphabet, we can write 20 different essential amino acid. Suppose we will take 1 or we will take singlet codon. So how many number of amino acid we can write? 4 amino acid we can write, but here we are having 20 essential amino acid. So we can take this singlet codon. Suppose we will take doublet codon. So how many number of essential amino acid we can write? We can write 16 essential amino acid, but we are having this 20 essential amino acid. So we can't write all the amino acid by using this doublet codon. So if we will take triplet codon, so how many number of triplet codon we can write by using this 4 alphabet? 4 into 4 into 4. 64 codon we can write, triplet codon. So this 64 codon will be much more enough to write these 20 different essential amino acid. So it is a system in which triplet codon of DNA can detect the amino acid of protein, and also it can specify which triplet codon for which amino acid. Genetic code having 6 different properties. First property is code is triplet. As already we have seen, if we will take singlet codon, we can write only 4 amino acid. If we will take doublet codon, we can write only 16 amino acid. If we will use triplet codon, we can write 64 different triplet codon. So by using 64 different codon, we can write 20 different essential amino acid. So this genetic code should be triplet. Second property is it is degenerate. Degenerate means if we take from this 64 codon, we can write 20 different essential amino acid. Remaining 44 triplet codon are also there. So those codon can also use to write those essential amino acid. It means for single amino acid more than one triplet codon can be used. So for single amino acid more than one triplet codon can be used. This is the degenerate properties of genetic code. Third properties of this genetic code is it is commaless. It means suppose here is the triplet codon for one amino acid. This is for one amino acid. Next triplet codon will for another amino acid. It means after one triplet codon, next triplet codon automatically will get synthesized next amino acid. There will be no gap in between two triplet codon. Fourth property of genetic code is it is non-overlapping. Non-overlapping means suppose this is the triplet codon for one amino acid. Any codon from this triplet codon can't use for other amino acid. Suppose from here it can't use here or it can't use for next amino acid. One triplet codon will use for only one amino acid. This will for one amino acid. Next triplet codon will for other amino acid. Fifth property of genetic code is it is non-ambiguous. It means there will be no ambiguity about a particular triplet codon. A particular triplet codon will always code for same amino acid. There may be degenerate. It means each amino acid may have more than one triplet codon, but each triplet codon will code for a particular amino acid. Sixth property is code is universal and it is always true in all type of cell present in this world. We can write this genetic code in this way. This is the first letter of triplet codon U, C, A, G. This will be the second letter of triplet codon U, C, A, G. This will be the last letter of triplet codon U, C, A, G. So how we can write U, U, U. Second will be the U, U, C. Third will be the U, U, A. Fourth will be the U, U, G. Similarly, here we can write C, U, U, C, U, C, C, U, A, C, U, G. Similarly, here we can write A, U, U, A, U, C, A, U, A, U, G. Similarly, here we can write G, U, U, G, U, C, G, U, A, G, U, G. In this way, we can write genetic code. So there are three different sequences. Those are known as stopping codon. Those sequences are UAG, UGA and UAA. These are known as stopping codon because they are not coding for any other amino acid. When any of these three triplet codon, one will be present in sequence. At that position protein synthesis will get stopped, or there will be no further protein synthesis will occur at that point. Similarly, protein synthesis will get start at a specific point when a specific codon will be there. Suppose in this mRNA, AUG is present. That time protein synthesis will start from there, because this AUG will act as initiation codon or initial codon for protein synthesis. When it will present at initial end of mRNA, then protein synthesis will occur from there onwards. Suppose this AUG will present in between in this mRNA. So, that time this AUG will code for methionine. When it will present at initial site or initial end of mRNA, that time it will act as initiation codon. If it will present in between, it will code for methionine. Similarly, another one triplet codon, that one is GUG, also sometimes act as initiation codon for protein synthesis when it will present at initial site, but normally it code for valine. If it will present in between in this mRNA, that time it will code for valine, but when it will present at initial end of this mRNA, that time there protein synthesis will get start. One variation you will find here, this UGA, which is acting as termination codon in universal genetic code. It will code for tryptophan or cysteine or solenocysteine in mitochondrial cell. In mitochondrial cell, you will find this will code for tryptophan. In different cell, it will code for cysteine and solenocysteine. This is all about genetic code.